Hello, everyone. This is Dr.Hira, and today we will talk about the enzyme catalysis and more detailed and more advanced topic. So previously we have talked about the why the enzymes can be the catalyst for the reactions, what are the impact and how the equilibrium do affect and how we can determine the different direction of the reaction. So now we will discuss and we'll see that how we can say the enzymes are the good catalysts, right? So enzymes are a great catalyst or the good catalyst because of the three main reasons. Whenever there is enzyme catalyzed reaction, so there are the three kind of things which enable enzymes, and these are the three events, the transient or the temporary events which are present, and the property which is making an enzyme a great catalyst. So there is the transient covalent bond between the enzyme and a substrate. There are the weak non-covalent interactions and the exquisite specificity. As we know, that enzymes are specific and they are very particular about the substrate and performing their action accordingly. So on the right side, you can see an energy diagram, but we will talk about this later. So at the moment, I have to emphasize that why we say the enzymes are a great catalyst, and they are great catalysts because of these three reasons. And these are because during the enzyme catalyzed reactions, there is the formation of a transient covalent bonds. They are the weak non-covalent interactions which are responsible for catalyzing the reactions when they are the enzymes and the exquisite specificity of that enzymes. So now we will discuss one by one about each of these three main reasons or the properties which enable the enzymes as the significant catalyst. So the very first thing is the transient covalent bond. So the transient name states that they are the temporary and they are the short-term covalent bonds, and these are the weak covalent bonds which formed during the interaction or the link between the enzyme and the substrate. So these transient covalent bonds are formed between chemical groups on the substrate and the chemical groups carried by amino acid on the enzymes in a site that is called active site where catalysis take place. So that is a straightforward thing. We all know that enzymes are made up of the proteins, and the protein's basic building block is amino acid. And in amino acid, we know they are distinct from each other on the basis of their R chains. Some are the anionic, cationic, hydrophobic, and the simple or the aromatic one. So the chemicals which we say as a substrate, so any of the compound chemical metabolite which is producing in your body system, or any of the drug which is interacting with the enzyme, so they interact through the chemical groups, the functionalities, the R side chains, right? So they are responsible for this, and we know that the enzymes are bigger in size. They are the uh, huge molecules, are macromolecules, but they do interact and they have the specific positions or the sites at which they can bind or link with the substrate, and they can perform their activities, and that is the active site. So these transient covalent bonds will activate the substrate, and in return, they will load the activation energy. So these transient covalent bonds are very important for making the enzymes as a significant catalyst, because if these weak covalent interactions are not forming between the substrate and the enzyme, so then the substrate is not activated for going into the transition state and eventually to the product one. So this link is somehow facilitating the substrate, and this is lowering the activation energy to which was the huge barrier if the substrate has to cover, and if there is no that kind of transient covalent bond or the weak covalent bond interaction between the enzyme and the substrate. So if they will be the separate ones, there is a huge hurdle or the energy barrier which a substrate has to cover. So this is facilitating substrate to activate and to lower the activation energy. From this figure, that formation of the transient covalent bond would be more understandable for you, that if you can see, this is an enzyme, and it is having a backbone, and we know that backbone is made up of the alpha carbon and the basic amino structure present in amino acid, and there are the certain R groups, right? So R side chains. So the substrate is basically, it is just showing you that this is some kind of a molecule which have the another cycle and, and another ring, just to show you that it has a two domain in a substrate. So when it interact, so whatever the group is feasible for this side chain to interact, right? So it will develop a covalent bond, because they should have the complementary um, groups which can interact with each other, and they can develop a transient covalent bond. So then, after this covalent intermediate, right? So this subject is activated, and then enzyme will perform its function, and this is converting this substrate into the product one. So the both are then released into the system. The next thing is the weak non-covalent bonds. The formation of each of these weak non-covalent bonds result in a release of a small amount of energy, and the summation of this small amount of energy form is called binding energy. So the people who are working in a computational chemistry and the people who are studying the biochemistry and the medicinal chemistry, they know the term the binding energy. So now we are explaining that actually what is the background of it, and why we say that this binding energy score is enabling us to give you the idea that either your that drug compound or molecule is good enough to bind, has a good binding energy with the enzymes. So that could be the potential for certain enzymatic or biological activity. So the binding energy that is released is the main source of energy that is responsible for the decrease of the activation energy. If that binding energy, or that amount of energy is not releasing when the substrate is engaged by the enzyme, and there is the transient a link or the friendship which is developed between the enzyme and the substrate, they are just making each other compatible with each other, so the enzyme can conduct its function on the substrate and help the substrate to convert into the product one. So this amount of energy which is released, so this is lowering that activation energy and making the reaction faster and feasible and viable. So through the reaction coordinate diagram, um, we can say that the binding energy is the difference between the gives free energy of uncatalyzed reaction and the catalyzed reaction. So I will show you the diagram in the next slide. So firstly, just discuss the outcomes and the significances of these weak non-covalent bonds. So usually the decrease of the activation energy by 5.7 kilojoule per mole results in the increase of the rate of the enzyme by 10-fold. So you can imagine that um, activity and efficiency, or the role of that enzyme binding energy release and with the enzyme function. So most of the enzyme will decrease the activation energy by 60 to 100 kilojoule per mole. The energy that is released by one weak non-covalent bond is in the range of 4 to 30 kilojoules per mole, which means that you need only a few of these weak non-covalent bonds to be formed for the catalysis to proceed. And that is an extremely important point. Because if we need the certain threshold number of non-covalent bonds to start the catalysis, to go through the enzyme action, so then that would be the harder one, so that would be the demanding one. But now the nature has created that uh, beautifully, that only the few of the weak non-covalent bonds can facilitate the enzyme reactions in a way that they can lower the activation energy to a much greater rate just with a few of the non-covalent bonds, and this catalysis can proceed with a very nice rate, right? So this is a diagram for you, and you can just see that this is a free energy G on an y-axis, and on an x-axis we have the reaction coordinates. And here you can see and you can compare. This is a substrate. It has its free energy, certain value of the free energy, and if it has to go to the uncatalyzed reaction, so this black curve is basically showing you the reaction which is uncatalyzed, where we are not using any enzyme, right? So it has to go to this barrier, this transition state, which is with the double steric, and then star. So then there is a product, and obviously the product has a lower free energy, so that is spontaneous, but the rate of the reaction will be determined that um, what how the enzymes are using or not, so how much time it will taking to cross that barrier, how difficult it is, right? So now if you see, uh, if we use an enzyme, so what an enzyme is doing is that there is the interaction between enzyme and a substrate. This is developing an enzyme and substrate intermediate, or you can say the enzyme substrate complex. Then there is a transition state, and then it is converting into the enzyme product complex, and eventually it's converting the product. Now you just can imagine, and this can compare what's the vast difference and how much um, greater reduction in the activation energy when we are using the enzyme. So the difference between this uncatalyzed reaction and the catalyzed reaction. So this amount of energy is releasing as a binding energy, and this is the lowering in that activation energy. So that what I have stated in a previous slide in an equation form. So for the convenience, I just mention it with the U and the C just to mention as uncatalyzed and catalyzed. But here I have it is properly written as an uncatalyzed or catalyzed one. This is another diagram for you to make it more visible and understandable for you. So just imagine we can just take an example, and we can consider that substrate is a metal stick we have on which that enzyme has to activate, and there is a certain kind of reaction has to proceed, right? So if our substrate is a metal stick and it has to break as a product into the two domain or the two parts, so what firstly it has to go, it has to bend, right? It has to change its conformation or the shape in a way that is visible and that it can easily go to that two parts of that stick, right? So this is a transition state. And if you just look at this energy diagram here, you can see this is a substrate, this is the product, and this is the amount of the activation energy which is required to for the substrate to go to these transition state and then eventually to the product one, right? And of course, that is much difficult to conduct at a faster rate, and obviously we need some external assistance for conducting these reactions. So now in a case of B, when enzyme is complementary to the substrate, what is in that case we have done is that this stick case, you can say, or we can use a term as a stick a staircase, like from the enzyme one, that we are taking that covering, right? And from the inner side, this there is a magnet, um, magnet coupled with that box, right, which is covering that one. And this is making complementary to the substrate one. Now it is very well organized, and there is a good complementary structure with enzyme and a substrate one, right? So what you can imagine is what can happen here, because it is a very much restricted domain, and this is just fit to that substrate one. So this is not enabling it to bend, right? So it means that this reaction is not viable. So what is and from the energy diagram you can compare and you can prove that one, that this is a substrate, and once it is in a complementary enzyme is in a complementary to the substrate one, so this is in a much stable form, right? It is even that much stable form that it has the much lower free energy of ES than the originally the substrate one. So it means that gap of the activation energy has further increased, right? For that transition state. So it means that this reaction is not possible. So we don't need that enzyme is should be complementary to the substrate. So what we need is that the enzyme complementary to the transition state, because that is the important. We have to facilitate for that transition state, which eventually has to go for the product formation. So if we are not facilitating this transition state, so then we cannot um, get the product one, right? So here you can see that this stick case provided with the magnet from the inner side, this is not as complementary to the substrate as you have seen in the B, right? So, but this is adjustable, and this is just complementary with the transition state. So this is facilitating the substrate that it get activated, and it can easily convert into the shape which is suitable and which is the most stable transition state. So now eventually there we will get the product one. And now if you compare the energy diagram, here you can see this is a substrate, and this is again the ES complex is not much lower and not much stable as the original, the initial substrate one. So then it has the less gap or the energy gap for its transition or the activation energy, right? So here you can see that can easily go to the product one. So in the case of B, you have seen that this was the amount of energy or the barrier the ES has to cover to the transition. And now this is much easier approachable for this ES to get into the transition and then eventually convert into the product one. So that delta GM is just the analog of the binding energy, right? Here, this is just in complementary to the magnetic property of this one, right? So we are taking this enzyme as a magnetic stickase, right? So you have seen the both all of these three cases and their relative energy diagrams, and what is the difference between if there is no enzyme, if it is uncatalyzed reaction, what if the enzyme is complementary only to the substrate one, it is not facilitating to the transition state, so this reaction is not going to happen. And if the enzyme is complementary to the transition state, so the perfect situation is this one, and that what we need is that enzyme is complementary to the transition state. Now, when the enzyme is complementary to the substrate, the ES complex is more stable and has less free energy in the ground state than the substrate alone. The result is an increase in the activation energy. That what I have um, discussed with you. So the third one is the specificity. And we all know that the enzymes are very specific in their reactions, right? And for this particular point, we will take an example of beta galactosidase. So the beta galactosidase is an enzyme which is involved in the hydrolysis of a family of the molecule which called the beta galactosides, right? So the beta galactosides is composed of a galactose unit linked to an R group that can have different chemical structure. That could be the aromatic, that could be anything. But the linkage between the galactose and R group is very specific bond called a beta glycosidic bond. And that is the bond that is cleaved in a reaction that is catalyzed by beta galactosidase to produce galactose and an R group. So beta galactosidase will function regardless of the identity or the chemical structure of the R group. That is not where the specificity lies. In fact, you can show the specificity by using an isomer. So what we do is that we have changed the isomer, and in one case, we have changed the alpha galactosidase, keeping the whole of the molecule the same one. And we then have examined that what happened. Either that enzyme is able to function on it or not, right? So in the next slide, you can see that one. So I can show you this. Here you can see this is the beta galactoside, right? And the enzyme responsible for carrying out this hydrolysis of OR by taking the water molecule, involving the water molecule into this OH, and as alcohol will be released because this hydroxyl will be contributed from the water molecule. Now, the beta galactosidase is an enzyme which is working on it. So, if you just look carefully on it, that this is the beta galactosidase site, right? And if we have another compound which is having all of the things same, and we are not changing the structure of this R, right? So, we are just changing this beta glycosidic bond to the alpha glycosidic bond. And you know that what the beta and the alpha glycosidic bond is, if this anomeric carbon OH is in the same position to this carbon 5CH2OH, then this is the beta one. If it is an opposite to that one, so that is the alpha one, right? So what happened when we use the beta galactosidase enzyme with the alpha glycosidic bond, though all of the structure was the same, so it should carry out, right? But it doesn't work. So there was no change of the galactoside into the galactose, or there was no removal of the R group, there was no hydrolysis carry on. So the reason was because this beta galactosidase, as a name state, it is very specific, and this is specific for this beta glycosidic bond, and this can carry out if this glycosidic bond is in a beta position. So then it can conduct it hydrolysis no matter what the R group is, right? So this small change prevent enzyme beta galactosidase to hydrolyze alpha galactoside. So this is basically confirming that enzymes are very specific for their substrate and for particular bonds, right? For the configuration of the molecules. So not so there the R group was not the consideration for the specificity. That was the beta and the alpha isomers, which where the specificity lies. Now the next is we will compare that what model actually prove this enzyme, and they just fit in and they have explained the enzyme or catalysis. So the very first one which we firstly had in our mind, and when we ever start talking about enzymes, so we straightforwardly go to the lock and key model, but that has its own explanation, and it has explained specificity of the enzymes very well, but that was not able to explain the efficiency of catalysis, because then it was not fitting well with that explanation. So this lock and key model was basically proposed by Emil Fischer in 1890, and he assumed that a substrate binds to the enzyme like a key in a lock, right? And you can imagine that that is a very restrict and a very strict model. So that can explain, okay, if there is a certain key that fit into the certain lock, so definitely. But how that catalysis is happening. So obviously, when we say, as you have seen in a previous lecture, that there is a conformational changes, there is the shape, there are the electrostatic interactions which are developing to facilitate substrate to the transition state and eventually to the product one. So if it is very strict and very hard, and there is a certain key and a lock, so it means we can't explain the catalysis well. So we can see that if we don't have an enzyme, as we have just taken example of the metal stick, and let's say we have a substrate with the two domains. So there is a transition state, it bends and then it's broken, right? So this was a reaction coordinate, and that is the activation energy. High activation energy, so negligible catalysis. So the reaction is just um, not happening, right? Now, if you compare the enzyme complementary to the substrate and enzyme complementary to the transition state, as I have just explained in the previous slide, so we are just taking up these with and linking up with the models, right? So if it is complementary to this one, so this is a very strict boundary, and this is not facilitating this substrate to the bending, right? Because this is just appropriate with the substrate shape. So it doesn't enhance the catalysis, because this electro enzyme and a substrate complex is so much stable that it has to gain a high amount of energy to cross that bond barrier to be at the transition state. So again, so there is the delta GB, which has to gain. So definitely that reaction is not going to happen, and this catalysis is not going to happen. Now, if we just compare this case, when this enzyme is complementary to the transition state, so this is facilitating this one, and this is very much happy with that enzyme, because this is facilitating it for conversion, for its transition to the transition state and then eventually to the product. So you can just take an example that if any of the institute, if any of the organization you're working in, or your environment is not facilitating through your transition period, when you are from the probation period to eventually to reach a certain level, so then it will be harder for you. You have to face a lot of frustration, you have to do a lot of extra efforts, and sometimes and mostly then you would not be able to show you that much productivity. So you that transition for acquiring certain skill, for be at certain level, so the environment, your organization, or whatever the factor is there, that has to be accommodated for you, and they should understand it, it should facilitate your transition state, right? So that is happening, that if the enzyme is complementary to transition states, this facilitating this one, and then the activation energy is much lower, and that activation energy difference, right, which was the high amount, and that was released to the binding energy, so that promote the catalysis. And how that binding energy releases, that we just have discussed through the non-covalent bonding interactions and the weak or the transient covalent bonds. The next model, which is a more updated one, and which was proposed after the lock and key model, was proposed by the Daniel Koshland in 1958. So he called this induced fit hypothesis. This model accounts for the deficiency of the Fisher model and is based on two assumptions. And these assumptions are very important, and I need your attention for this. So, the first assumption is that you have optimum weak interaction between the enzyme and the transition state. The second was the second assumption is that upon binding of the substrate to the enzyme change conformation, and that is the induced fit that optimize the position of their reacting groups and favor enzyme catalysis, right? But and these two assumptions are the statement were missing in the lock and key model, because that was just a very strict model that it was just saying that an enzyme is just appropriate and fit to the shape of the substrate. So this was not explaining that how the catalysis will conduct when there is the changes in the conformation. So what is happening with that enzyme? So that was explained by the induced fit hypothesis. So, in conclusion, what we extract from this discussion is that we have seen that we have weak non covalent interactions that contribute to a decrease of the activation energy as well as the specificity of the enzyme. We have transient covalent bonds that are formed that decrease the entropy of the system and consequently favor enzyme catalysis. And then we have these transient covalent bonds also contribute to the correct positioning of the reacting group. So there is another aspect which is the desolvation. In the cell, a substrate is surrounded by molecules of water. So this is not a static environment when we are talking about the biological molecules, when we're talking about the cellular metabolism. So there are number of water molecules, million of water molecules which are present there. So, and there is formation of weak interaction between the water molecule and the substrate. Now, the dissolution is in fact the process by which this weak interaction between water and substrate are replaced by weak interactions between the substrate and the enzyme. And this weak interaction will contribute to the binding energy and will facilitate catalysis. So, in conclusion, catalysis is driven by the structure and the conformational changes. And these conformational changes are explained by the induced fit model. And you have also seen that the complementary attitude of an enzyme towards its transition state is a key for the process of catalysis. So that's for this enzyme catalysis topic. And I hope you get that point and you are able to have a lot of answers, but you may not have think about that how the enzymes are conducting their catalysis. So if you further have any queries, so just comment on a video or you can just email me. So I would try my best to answer your queries. Thank you so much. Have a good day.